Hey, good afternoon, everyone. I'm very excited here to share with you my experience with the phi mapping approach. So without further ado, let's dive right into the phi mapping. So first of all, why we want to do phi mapping? We know GWAS has been very successful in finding a huge number of associations with complex traits and complex disorders. Take the Crohn's disease, for example. Yeah, the cursor works, great. Take the Crohn's disease, for example. Before 2005, we don't know a lot of genetic factors underlying the Crohn's disorders, but within the past 10 years, the knowledge for the genetic association for Crohn's disease grow exponentially, and we now have hundreds of loci underlying this disorder, and we are very familiar with schizophrenia. Before GWAS year, we don't really have a very conclusive genetic factor associated with schizophrenia, and now we have over 200 from the PGC. So in summary, GWAS has been extremely helpful in giving you a large number of disease associations. However, what GWAS wasn't able to give you was the mechanism underlying these associations. And we see that this doesn't look right, because if you take a very intuitive, a very naive look at these GWAS loci, you will see that many GWAS loci actually implicate region with at least one gene. So for schizophrenia, and I just got the number, is 75%. So although most of the top associated GWAS SNP are non-coding, we do see that 75% of the GWAS loci in schizophrenia are next to a gene. So, and second, we saw that many GWAS associations are in linkage disequilibrium with the coding variation. For schizophrenia, this number is 10 out of the 108. So we also see that many GWAS loci co-localize with functional elements such as expression QTL, histone marks, so on and so forth. So really, it's no reason why the GWAS give us so little insight into the functional mechanism underlying the disorders. But in fact, if we took the best studied disease, the IBD, the inflammatory bowel disease, you will see that really we only have a very handful of loci that we know what is the causal gene and what is the causal mechanism. So what phi mapping does is actually a three-step study to help us to understand the mechanism of the GWAS locus. So the first step is to understand what variants are really involved in the disease process and what biological functions these variants disrupt. And lastly, what is the disease-causing mechanisms. So what is causing trouble for GWAS study to tell us the causal mechanism? The trouble is actually caused by something called linkage disequilibrium. Even across different genes. So how do we deal with this? So in phi mapping, people typically use an approach called the Bayesian approach. And this is different from the hypothesis testing framework, which we use the frequentist approach generally to describe them. So in the following slides, I'll give you a very general introduction in the difference between the two approaches, and I will show you why the Bayesian approach is in general preferred in phi mapping. So let's use a toy example to understand this. So suppose that you're a Marine officer and you want to identify where this submarine is. So you have a sonar device here that can help you to tell roughly what the location of this submarine is. So by using this sonar device, if you are a frequentist person, you use a frequentist approach, what you will do is you will divide the ocean into different subregions. And for each of the region, you will test the signal you obtained for that region, and you'll see whether the signal you obtained is far beyond the signal under the null, which is the signal that there's no submarine. So if you see a very strong signal that is beyond, way beyond the null, beyond the signal there's no submarine, you say that this region is associated is significantly associated, it's more likely this region has a submarine. And you do that for every little region, every little division in the ocean, and this is the result you have. You have an area that is significantly associated, which means they have significant signal deviation from the null, and you have regions that are less significant or non-significantly associated. So this is what the frequentist approach will tell you. However, there are a couple approaches. So what you have here by using the frequentist approach is a p-value. And the p-value literally tells you how likely you're wrong by rejecting the null. It's not a probability, or it's not a probability that you would expect. It doesn't tell you how likely the submarine is here. It's not very intuitive to understand. And also, it doesn't sum up to be 1. So that's one of the key issues. And it doesn't have any prior information. So say that we know there's only 1 submarine in the ocean, the approach won't be able to account for that information. It doesn't give you the condition probability that there's 1 submarine in the ocean. So what the Bayesian approach does is something more sophisticated. You can combine various evidence, different data in a Bayesian approach. So in this example, let's see, we have the information from sonar that this area is likely to be implicated. It's likely to contain the submarine you're looking for. And we also have something called prior knowledge. So as a marine officer, you know that there's a range, there's a depth of the submarine they usually swim at. So by combining the two approaches using something called Bayesian's formula here, you are able to have a more sophisticated description of the region. So what you end up with is, by combining the two different evidences, for each of the small region, the division in the ocean, you're able to have a probability, and that is a real probability, how likely the submarine is in this location, and that's going to sum up to be 1. All right, so that's a toy example for the difference between Bayesian's approach and the frequentist approach. And let's see how it applies in a real genetic study. So this is a genetic association plot called the regional association plot. I assume everyone here have seen this before. So what it does is it tests for every genetic variation to see whether it is associated with a disease or not. And what you have here is a p-value association. And we have a genome-wide significance threshold. Anything beyond that threshold is believed to be significantly associated, and anything below that threshold is believed not. So this is what you get by using the traditional GWAS. You have a bunch of things beyond the threshold, but it doesn't tell you which one is the causal variant and which one, and what mechanism they implicate. So to help us to better understand the example, I have to give you a little information about the disorders. So the disorder we use here is called the inflammatory bowel disease. It has two etiologically related phenotypes called the Crohn's disease and the ulcerative colitis. The Crohn's disease can happen anywhere in the body, including blood and the GI tract, and the ulcerative colitis is restricted to the colon. All right. So what fine mapping does is it will organize the very complex GWAS regional association plot into very simple interpretable tracks. So this is the tracks they map all the genetic association into. So first, the GWAS, the fine mapping algorithm is able to find there are 2 independent associations for this region. The first association is only to the Crohn's disorder, and the second association is to both Crohn's and the ulcerative colitis. And we use IBD here to label both disorders. So the first association is the association with the Crohn's disease, and fine mapping is able to tell you what are the variants that are likely to be accountable for this signal. And in this example, we have 5 different variations that are likely to be accountable for this association. And for each of the SNP, we have a probability. For example, the first one has 40% probability to be causal, and the last one has 8% probability to be causal. And of course, for each of the SNP, we can annotate their function. We have the first SNP disrupting a transcription factor binding site, and the last SNP disrupting a epigenomic mark. The second signal is even more cleaner. It was mapped to a single variant with more than 99% probability, and that's a coding variation, which is a very beautiful explanation for the mechanism. So that's essentially how the output of a fine mapping study. So how do we do fine mapping? I show here that we'll need 4 essential things to happen for a fine mapping study. The first thing we need is a large enough sample. We all know that GWAS takes a large sample to work. It needs a sample large enough to have enough power to find the genomic loci. But for fine mapping, actually, it takes more samples, unfortunately. So this is a power illustration comparing the power for GWAS and the power for fine mapping. So the dashed line is the power for GWAS, and you can see that if you are looking for something with odds ratio of 1.2, allele frequency of 10%, and equal case control ratio, and if you have 15,000 samples, you are almost certain to be able to find this locus in GWAS. However, if you do fine mapping on the same locus, assuming there are 2 variants in this linkage disequilibrium with R² of 0.8, you only have 50% power. And if the R² of the linkage disequilibrium increase to 90%, to 0.9, or even to 0.95, you basically have no power. You have less than 20% power to do that. So you can see that it's more challenging to distinguish 2 variants in this linkage disequilibrium. It just needs more samples to have the power to do that. And imagine that for most of the loci that we're dealing with, you don't just have 2 variants in high linkage disequilibrium. You usually have more than 2, 5 or 10, or even several, or even 20 variants. So it makes it very challenging to do that. But luckily, the many international consortiums have studied many, many years earlier to put together all their data, which can be very helpful for the fine mapping activity. And I'm showing here the International IBD Genomics Consortium, which has put together more than 75,000 cases and 35,000 cases and a similar number of controls, which we were able to leverage to do the fine mapping study. And for the PGC, the Psychiatric Genomics Consortium, which everyone here is very familiar with, they have done a great job in putting together over 35,000 cases and 100,000 controls in the past years. So this did give us a very good sample set to play with for the fine mapping study. The next thing we need is a good genomic coverage. So talking about genomic coverage, the first thing that came to your mind is probably the whole genome sequencing. That is indeed the best technology for achieving a good genomic coverage. However, it is still sort of expensive comparing with the genotyping chip. And remember, I told you that we'll need to do that in a huge sample, which makes the cost very inhibitive if we really want to use the whole genome sequencing approach. The exome sequencing approach is very attractive in that it has a good coverage in the coding region. However, a lot of the GWAS happen outside of the coding region. So unfortunately, it's not a technology that is the best fitted for fine mapping. Some studies would add the exome sequencing to the existing data to achieve a better resolution in the coding region, but it cannot be used by itself for the fine mapping purpose. So what's the most commonly used approach is the genotyping chip plus the imputation approach. We say that not all the chips are created equal. Some of the chips, especially the early days chips, they have too loose coverage of the genome. They're probably not the best for fine mapping. So what is good for fine mapping? The ideal chip design for fine mapping is the chip that with the genome-wide coverage, so that you can use the genome-wide coverage to calculate the principal components for population correctification. And they also have a high density coverage in the area that you are very interested in. So examples for such chips are the psych chip for psychiatry-related loci, the immunochip for autoimmune-related disorders, and for metabo chip for metabolic-related disorders. So for this chip, because they have specifically designed probes in the disease-related region to increase the density, it gives you an edge in the genotyping quality in the after imputation to capture the variants in the region. So how well are we doing by using this chip plus imputation technology? So this figure gives you an idea how well we are doing. So we break the variance to, based on the allele frequency, because that's the major factor. The allele frequency is a major factor deciding whether you get a good imputation or not. For the extremely common variants, for variants with more than 5% allele frequency, you can see that you can almost impute everything perfectly. So you have excellent coverage for this region. For variants that have between 1% and 5% allele frequency, you can see that most of them have good imputation coverage. I think the number is you have more than 95% captured with more than 0.8 quality score, which is a decent number. So the conclusion here is for more than 1% allele frequency, you can do a reasonably good job. But for the variants that are rare or or of low frequency with less than 1% allele frequency, really you don't have a lot, imputation quality out of it. So I'm aware that recently there's a type 2 diabetes paper, file mapping came out, which claims that by using the HRC reference panel, which is a huge, much bigger reference panel as I used in this slide, as well as a huge study sample, you can probably achieve a accurate enough, complete enough coverage for variant with down to 0.1%, 1 magnitude below this allele frequency. But I haven't really tested myself, and I still think 1% or at most 1.1%. That's probably the best you can do with a chip plus imputation approach. The third thing you need is the quality control. Quality control is extremely important in file mapping because for GWAS, the quality control is actually easier because you only need one variant to be associated to the disease in a locus to say the full locus is relevant to the disorder. But for file mapping, you want to find the causal variant, which asks you to evaluate every variant carefully. And because of that, you cannot be as generous as the GWAS study, and you have to be very cautious in your QC. You don't want to throw away too many SNPs, which could very well contain the causal SNP. You want to be strict enough to remove all the false positives, but don't over-strict to remove the useful SNPs, which makes a very challenging task for file mapping. So for file mapping, there are basically two flavors. One is called the mega-analysis, and the other is called the meta-analysis. And we're going to review the pros and cons for both of them in terms of the quality control. For the mega-analysis, we basically put all the raw genotypes together for one study. And the benefit is once it's done, it's very easy to use the data set. You have the raw individual level genotype for all your samples, and you can basically run every mathematical model, your favorite model, on this data set. And also, you have better power because you will have reduced power if you have a case control imbalance in different cohorts. But by putting everything together, you are able to balance the case control ratio, which gives you a better power. However, the disadvantage of this approach is you need to do extensive QC for this kind of approach because you are putting all the data together. It's very dangerous there, if there are heterogeneity remaining in the data set. You have to make sure you have to do extensive QC to remove potential heterogeneity. And also, it has a very high demand on the computational resource because you do have the individual level genotype. That's the number of samples times the number of variants, which is a huge number. Just to give you some flavor, some taste how the QC is going to happen if you do put all the data together. So this is the QC we used in a real analysis. This line is all the typical GWAS QC. And in addition to that, we did heterogeneity test to remove the potentially heterogeneity across platforms. And we do the imputation after that. And we do the manual cluster plot inspection to make sure nothing, significant heterogeneity and genotyping error exists in the data set. And what we did was we redo everything after we do that, and we redo the imputation to remove the contribution from the false positives. And we do this iteratively until we are certain there is no false positive remaining. So this is a very tedious work if you really want to do that. And this is how we do the manual cluster plot inspection here. We just use, we eyeball the result to capture the faulty batches. The second way of handling the data is to do a meta-analysis. The meta-analysis basically does the association test in each of the sample independently and then combine everything through a meta-analysis. The benefit of that, the advantage of that is you don't have to be very strict in quality control because very likely the genotyping error or whatever error will likely to happen in a particular cohort. It doesn't it's very unlikely the same error keep happening in different cohort, in different countries, in different genotyping technology. So by doing a meta-analysis, you borrow the information from other cohort to remove the error from one cohort. And also it's less computational intensive because instead of having 100,000 individuals, you only have one set of the summary statistic. It only grows linearly with the number of variants. It doesn't grow with the number of samples. The disadvantage is of course, you have very limited numbers of statistical tests you can use. Everything has to be either an approximation or it has to be under the assumption of a linear model. So there's limited toys you can apply to this data set. And also, it's very challenging to model the linkage disequilibrium very accurately because most likely you will have the linkage disequilibrium from the southern genome or from the HRC, which may not be the best representation of the linkage disequilibrium for your own study cohort. The last thing we need is the fine mapping algorithm. So there's a very nice review paper discussing all the fine mapping algorithm out there. I'm not going to go through every detail of the paper, but I do want to give you a very general introduction in how the methods differ from each other. And I promise this is only a slide with mathematical symbols on it. So basically what you do is you want to calculate the probability of a set of SNP, whether they are causal, given all the data. So this is a very abstract description of the fine mapping, how it works. So you want to know whether, given a set of the SNP, whether they are causal or not. You calculate the probability for that. And you use the base symbol, you use the base formula for that, which can be derived mathematically to this equation. And there are three terms in the equation. The term as a denominator is very simple. It's simply a normalization constant. It makes sure the probability sums up to be 1, so nothing fancy. You don't need to worry about that. And the two other terms are the prior term and the likelihood term. So they are really the meat of the fine mapping approach. And basically, all the Bayesian flavored fine mapping approach differ in how they handle the two terms. So the first term is the prior of the SNP that are causal. So for the prior, you can assume every SNP to have the same prior, which is a very simple and non-informative prior. Or you can incorporate the functional annotation. You give SNP that are coding a higher weight, for example, or you can learn the weight from some fancy algorithm. So that's a big difference. For many fine mapping algorithms, they give different weights to different SNP, and how they give the weights, how they calculate the weights is a field of development. And this term is the likelihood term of the data given the causal SNP. And different fine mapping algorithms differ in that what phenotype they can take. Some of them only handle the quantitative trait. Some of them can do the binomial trait, the case control trait, and some of them can do the multinomial trait. So that's one major difference across all the methods. And also, some of the approach is able to run the algorithm only on the summary statistics, and the rest of the algorithm may need you to provide individual level genotype data. So that is also a difference, across the algorithms. And how you model the likelihood function, the probability of data given the causal SNP is also a field of development, because very likely you're going to need approximations to speed up, and people would assume different mathematical model because they view the genetic architecture for disease differently. So the algorithms may differ in how they model this function as well. And the last thing that can differ across the algorithm is how you choose the coding SNP. So I put it very abstractively as causal SNP here, but as you can imagine, a genetic locus can have thousands of SNPs. In order to calculate this probability, you have to iterate through all the possible combination of SNPs, which is an impossible thing to do. So different algorithms would use different strategy, and some of them allow there are multiple causal SNP, and some only allow a single causal SNP or a fixed number of causal SNP that's known prior. And some algorithm would use a stochastic approach, which means they will randomly test different SNP pairs, and some of them would use greedy approach to increase the speed. So that's basically how all the algorithms differ with respect to each other. So I'm not going through the details, but as you can see, the algorithms differ in terms of the trait, in terms of whether they are using the summary statistics, whether you need to tell them in advance how many causal SNPs are there, whether they are using the functional annotation, whether they're searching the SNP through the MCMC, which is a stochastic approach, or through the greedy approach, or through the exhaustive approach. So I think that could help you with how, to understand the difference between the algorithm and how to choose the algorithm that best fit your own study. So I'll focus on the algorithm that I designed myself, which is conveniently called fine mapping. So I'll just give you a very intuitive idea how it works as an example. So it actually works very intuitively. So in a genetic locus reported to be associated with the complex disorder, this algorithm finds the SNP and phenotype pair that has the best log likelihood, like this. And then it grows this pair by adding either a new SNP, so you have two independent SNPs associated with the same phenotype, or it adds a new phenotype to it, so that you have a single SNP controlling two related but different phenotypes. Or it can add both a SNP and a phenotype, meaning that in this locus, you happen to have two SNPs, and each moderating one phenotype. And you choose out of the three possible outcomes which one is the best one, and you pick up the best one, and you keep growing this model, so on and so forth, until the model doesn't grow anymore. So that's essentially how this method works. So for the interest of time, I'm going to dive right into the result. So we use the inflammatory bowel disease as a result. So this is the outcome of the fine mapping when it's applied on the inflammatory bowel disease locus. So as you can see, we started with 94 locus because although we have 200 of them, not every locus is powerful enough for the fine mapping study. So we chose the 94 that have sufficient power. We did the fine mapping on it. We found that 68 of these locus host only a single association, and the remaining of them host more than one association. And that results in 139 independent association out of these 94 locus. And we ran fine mapping on this 139 genetic loci, and we found we were able to map 18 out of this 139 genetic association to a single causal variant, which is a big improvement from the GWAS locus. And this is the 18 genetic associations that we were able to map to a single SNP. And I'm just going to show you one example, which is the NOD2 gene. In this gene, we are able to map the previously reported 3 coding variants using the sequencing study, and now we are able to recapture it using the CHIP plus imputation study. And we're able to capture additional ones shown in the box in a slightly larger, not in one single SNP credible set, but in 2 or 3 SNP credible set. So this is a big improvement. We no longer need to do the exome sequencing. By simply using the chip plus imputation, we're able to capture a lot of the causal signals. So what if we zoom out a little bit to look at the global distribution, the overall functional landscape of the fine-mapped causal SNP? What do we get? The first thing we noticed was an enrichment of the coding variation. So we saw, comparing with the silent, with a non-coding synonymous variation, we saw a more than 20-fold enrichment, which is a big, huge enrichment. And I do want to point out here that this, you shouldn't interpret this as the coding mechanism is a dominating mechanism for IBD, because as I will show later, the coding variation have a bigger effect size. It's just impact the disease to a much bigger extent so that you have better power to pick them up first. It's very likely that the non-coding variation, although they have a smaller odds ratio, they have less power to be captured here. They still contribute significantly to the disorder. So this shouldn't be interpreted that the coding variation is more important than the rest. It simply means we have better power to pick up this coding variation. We then apply the fine mapping to the, we then look into the transcription factor binding sites, and we saw a modest enrichment comparing with the synonymous variation. So we saw 3 variants associated with IBD mapped to more than 50% probability. They disrupt the transcription factor binding site. And this is one of them, which is very interesting in that it's highly conserved across all the mammals, I think, from dog to mouse to chicken to human, highly conserved. And we're very lucky in that the encode cell line, the HeLa cell line in encode, is actually heterogeneous in this SNP. So we were able to pull the chip-seq data out of the encode cell line, and we saw that the wild-type motif have very high binding affinity to the AP1 transcription factor. But when the for the variant motif, the binding affinity is almost reduced to none. So this is indeed the function that's being interrupted by this variation. So the next thing we looked at is the chromatin peaks. This is a little more complex than the coding variation and the transcription factor binding size I showed you, because it depends on different tissues, and we have various histone marks for this analysis. So what we did was a very simple reduction. We took every tissue and every transcription factor, and every histone modification marks, and we see for each combination of them, do we see enrichment like we saw before for the coding and for the transcription factor binding site. And for each of them, we test whether they are enriched, and we give the color based on the enrichment, and we organize the tissues into the immune-related tissues, the GI-related tissues, and other tissues. So without any fancier algorithm, you can simply tell by your eyeballing that the histone K4 methylation 1 is enriched in the immune tissues, and the K27 acetylation is enriched in the GI tissues by simply looking at the color. So what we did was we took this part of the data. We took the immune tissues for K4 methylation 1, and we took the K27 acetylation marks for the GI tissues, and we did a similar analysis as we did for the coding and for the transcription factor binding site. And we indeed saw enrichment in these tissues for these histone marks. So lastly, what we did was to look at the EQTL, the connection between the expression quantitative loci with respect to the fine-mapped genetic association for IBD. And there are several previous reports that the EQTL may be driving a substantial proportion of the disease genetic associations. And we looked into these reports, and we found that they didn't do 2 things. So first, they didn't use fine mapping. So they took a very big and pervasive genetic association instead of using a more precise and fine-mapped genetic association. And the second, they didn't account for the coincidental overlap between the EQTL and the genetic association. And we say that this is important because EQTL is very abundant in the genome. So this is a toy analysis we did. We looked at every 50 kb. We looked at 50 kb of every transcription starting site, and we found that 80% of the time, if you look into the 50 kb of the transcription starting site, you are able to find a eQTL. So this is very pervasive. It is basically everywhere. You have to adjust for the coincidental overlap between the two things. So what if you do? So what if you use a phi mapping and you adjust for the coincidental overlap? We did that for 2 independent data sets. The first one is a twin study with 2,752 twins. We found that by using a phi-mapped disease association, we saw 3 associations overlapping the eQTL. But by expectation, we would expect 3.7. So this is really not enrichment. We don't have a major eQTL signal accounting for the disease association. And similarly, for another independent study of 8,000 individuals, we don't see a significant enrichment of the eQTLs explaining the genetic signals. However, when we turn into a smaller but more tissue-specific data set, we do see some moderate enrichment signal. So this data set was generated only using 100 to 400 samples. It's much smaller than the studies I showed you. But as you can see, by using this cell line specific data, we're starting to see moderate enrichment. The p-value is significant at 0.01 and 0.018 for the CD4 and for the ileum tissues. So basically, what it tells us is we need to have an understanding of the non-coding genome in a tissue-specific context in order to understand the genetics association. It's not just the genetic, it's not pointing us to the right signal. We do have the right signals and the phi-mapped signals from the genetic studies if we don't understand the non-coding genome enough to interpret how these genetic signals work and what it tells us. So in the remaining time, I'm going to extend a little bit into the psychiatric genomics study, as I think that's the interest of the audience. And I'm going to introduce a little bit about a phi mapping algorithm that I'm actively working on. So as I showed you, the coding is actually driving the phi-mapped signal in the IBD. What I did here is to look at the distribution of the odds ratio for the IBD and for the schizophrenia signal. As you can see, the IBD signals are indeed driven by the coding variation, and the coding variation have a huge odds ratio. Even for the non-coding variation in the IBD genetic association, they still have a very large odds ratio comparing with the schizophrenia association. So that's the reason that we're able to do a good job in the IBD in the IBD phi mapping. We're able to map a lot of them to single SNP credible set. But this would be much more challenging for the schizophrenia genetic associations because they are much less powered. They have less effect size comparing with the IBD ones. So what can we do? Of course, increasing the sample size would be one idea, but there are a much smarter way to do that. As I showed you earlier, the challenge for phi mapping really comes from the linkage disequilibrium. The SNPs are in high correlation with each other, causing causing this to be very hard to tell which SNP is the causal SNP. But we know that the linkage disequilibrium can be different in different human populations. For example, the pattern for the Caucasians can be very different from the pattern in the Chinese and Japanese, and very different from the patterns in the African populations. So we can actually leverage this difference in the population genomics data to improve our phi mapping resolution. And this is how it works. See that if you have 3 variants in high linkage disequilibrium with each other in the European population, and this is the causal SNP. By only using the European data, there's no way you can tell which of the 3 is the causal variant because they are 100% correlated. There's no way you can tell. But if you bring in the East Asian data and assume the East Asian data have different population structure, population linkage disequilibrium comparing with the European data, you are able to see that only these 2 SNPs are correlated because the LD is different. And under the assumption that the causal variant is shared across populations, which I will show a little later, you will see that only the causal variant have strong association in both populations and have little heterogeneity. So non-causal variants, because they're tagging the causals through linkage disequilibrium, they're likely to have strong association in one but not both population. And if you test the heterogeneity, they're going to show some degree of heterogeneity because of the linkage disequilibrium. So luckily, in schizophrenia, we have a large number of East Asian investigators that have done great work in recruiting a large number of samples. So we were able to put together 13,000 cases and 16,000 controls for the schizophrenia data with East Asian ancestry. So first of all, I'd like to know whether the causal variants are really shared, because that's important for this algorithm to work. So naturally, you can expect if the causal variants are indeed shared, you would expect heritability to be similar across the two populations. So that's the first check we did. And indeed, we saw that the heritability are very similar to each other, is very close to 0.24 in both populations. And the next thing we did was to look at the genome-wide genetic correlation between the two populations. And we adjusted the allele frequency and adjusted the population genomic difference. What we got is a very now is a number very close to 1. We got 0.98 with confidence interval between 0.95 and 1. So the genetic correlation is basically telling us we do have very similar genetic effect across variants in the two populations. The last thing I did was a little bit challenging. I did a direct comparison of the odds ratio from the European and from the East Asian population. I say this is a very challenging comparison because the Europeans' data is 3 times larger than the East Asian data. So the Europeans are basically driving all of these loci. And because of that, there's a winner's curse in the European data, and that's why you see a shift towards the odds ratio in the European direction. But nevertheless, the directions of this association is consistent across all the loci, and if you consider the winner's curse, this deviation is actually not very bad, and we have a statistical method that corrected the winner's curse, and after the correction, it shows a slope that is indistinguishable from 1. So basically what it tells us is you can trust what I told you, the genetic effects are indeed very consistent across the two populations, for schizophrenia at least. Oops. So I applied the algorithm to a locus from schizophrenia as an example. So this is before the phi mapping, the trans-ethnicity phi mapping. You can see the two SNPs are highly correlated, and there's a bunch of SNPs with very close p-value. So when I apply the phi mapping approach, the two SNPs are immediately distinguishable, with the rest of the SNP have much worse p-value. And this is exactly as I showed you. This is by only using the European data. You have everything in linkage disequilibrium, and you have things in very significant p-value, very close to each other. But once you add the East Asian data, you can see that the linkage disequilibrium is getting much better, and only the top SNP here shows good p-value significance and no heterogeneity across the SNPs. So that's essentially how it works. When you apply this to the genome-wide data, you can see that a lot of the loci have a reduced number of the causal variants. We have twice the number of loci that's been mapped to less than 5 SNPs. And in general, every genetic loci have less SNP once being phi mapped using this approach. Unfortunately, what I cannot show you is a genome-wide analysis like I showed you for IBD, because the data is just not there yet. The hope is when the Stanley Center Global bring in additional samples from East Asia, from Africa, and from Mexico, we're able to have a much larger data set, much diverse data set, especially the samples from Africa, which has a smaller linkage disequilibrium, smaller LD blocks that can help us to further map these genetic associations. So in summary, I hope I've convinced you that the phi mapping can be done using the ultra-high density genotyping array with imputation. It can help us to understand the function of these genetic associations, but we need more knowledge of the non-coding genome to understand the function. And also, the population diversity can really help us to do a better job in phi mapping. And thanks, everyone. That's all.
Thank you. Thanks for putting together a really exciting session, and I think the talks complement each other well. They're sort of both thinking about what we do with GWAS data, how we go forward from there. And the previous talk was focusing on fine mapping the genetic variants, and I'm going to be talking about identifying the downstream functional targets and really the target genes at GWAS loci. So the motivation is, you've performed a GWAS study. Let me plug this in. You've identified genome-wide significant variants, and you see these sort of local Manhattan plots with significant associations in a really large number of genes. And what do you do next? How do you identify the genes that are likely for experimental follow-up, or how do you identify genes for then taking them into pathway models or sort of better understanding the real etiology of the trait? And one thing you can do is you can just sort of Google these and hope you get lucky, and I've done that in the past. But what we're trying to develop, or what we've sort of proposed, is a method that uses EQTLs and transcriptomics and other functional data to provide scores for these genes and allow you to rank them in a more computational way. And the motivation for that is, of course, there's been a lot of work on mapping genetic variants to the transcriptome through EQTL studies in the GTEx here, and as well as other large consortia, including the PsycOncode, for example, or the Common Mind. And they have converged on some initial evidence that these EQTLs, or genetic variants associated with expression, are also enriched for genetic variants that are associated with disease. And I think actually, as was mentioned in the previous talk, a lot of this matters, a lot of this depends on how you account for the sort of background disease architecture, which there's a lot of different ways to do. But in general, it looks like from this paper of Hormozdiari et al. Last year in Nature Genetics, there was this very significant, perhaps modest enrichment of EQTLs, fine-mapped EQTLs for general disease heritability from common traits. And in that analysis from Gamazan et al., using the GTEx data, again, looking at the fraction of disease heritability that's explained across a variety of traits here on the X, we see a significant and fairly substantial total fraction of heritability explained. We also see, perhaps peculiarly, a very relatively small contribution of tissue-specific effects, although this is probably due to power and not having sufficient sample size to really effectively identify them. But so there's some evidence already, just genome-wide, that EQTLs are relevant for disease architecture. And what I'm going to be talking about is leveraging this relationship to now identify target genes. So first I'll talk about how we do this through the concept of prediction, how we apply these methods to GWAS data where we only have summary statistics and we don't have individual level data, and then I'll talk about some sort of ongoing and future directions. So in principle, if the goal is to connect SNPs to, that are associated with disease to a target gene, one way, or maybe the optimal way that we would do this, is we would take our GWAS cohort where we have the genetics and the phenotype, and we would measure expression in every single individual, in tens of thousands of individuals, and then we could sort of interrogate this this association directly. We could look at whether expression is associated with disease. This is like a differential expression analysis. We could also look at whether there's genetic correlation between the expression and disease. And we would do that for every single gene, and it would tell us which genes are the most strongly genetically associated with the trait and are therefore sort of the most likely causal candidates, although this is still always in the realm of association. The problem, of course, is that we usually don't have data like this in sufficient scale to be able to kind of leverage effectively. So typically what we have is gene expression studies that have been performed in a small number of individuals, in GTEx, for example, in a large number of tissues, but still in a relatively small number of samples. And then disease GWAS data that has been collected in extremely large populations, now with the UK Biobank or with the Giant going up to, I think, over 1 million individuals. And so the goal is to be able to do this, or something close to it, with data that's sort of in these two different silos. And the way that we do that is basically through prediction. So there's been a lot of work on polygenic risk scores and predicting complex phenotypes into individuals. And what I'm talking about here is predicting molecular phenotypes instead, and specifically the cis component of molecular phenotypes, which is sort of where the EQTLs are also coming from. And so the idea is that we take this reference panel of expression for which we have individual level data, and we can build genetic predictors of the expression for every single gene within every single tissue, and we get sort of a weighted score of the relationship between these variants and the transcriptional activity. And then we go into our cohort with just the disease and the genetics. We do the prediction, predict the molecular phenotype, the predicted expression, and then we can perform something akin to that previous genetic correlation analysis, but now we're testing the association between predicted expression and the phenotype in this cohort. So prediction basically gets us around the fact that we've really, we measured something over here that we've trained, and we measured something over here that we're testing into. And this has actually been shown to work quite well in a number of papers. So we applied some prediction methods in this paper from 2016 compared to across a couple of cohorts, and then there was work from Gamazan et al. In 2015, also looking at prediction of gene expression. And the general picture is the same, which is that, first of all, the top EQTL, the lead SNP, explains a lot of the variance of this of the cis component of expression. So this actually kind of shocked us initially, but it looks like the top EQTL explains about 70% of the local cis heritability of an average gene. But by leveraging all of the genetic data in the locus and by building this more complex prediction, you can substantially increase the predictive accuracy by about 1.3-fold on average, and you kind of see that in the difference between these bars or in the difference here between top SNP and an elastic net prediction model. And then there's a tail of genes for which you can do a lot better, which most likely have multiple causal variants, and so integrating them all together does you better than looking at a single SNP at a time. And in general, the other kind of positive thing is that as you increase in sample size, you start to see more of these subtle secondary effects, and you're actually able to improve the prediction further and further within larger studies. So this is the idea at the individual level. The next and sort of primary aim is to be able to do this when you don't have individual level disease data. So a typical scenario is we have a reference panel of expression and we've trained our complex predictive models, but then the target data that we have is just GWAS summary statistics, so z-scores and p-values or betas and standard errors, and then perhaps a reference panel of LD. And what we've developed is a method to be able to, instead of doing the prediction directly, to be able to estimate the association that you would have seen if you did the prediction directly using the reference data and just the GWAS z-scores. And to give you some intuition for how that works, one way to think about it is imagine there's a world in which there's no LD between SNPs, and you performed a GWAS analysis, and you see some SNPs are strongly associated with the trait, and everything else is null. And then you have a missing SNP that wasn't typed on your panel, but you want to know what the association for that missing SNP is. And for us, the analog to that missing SNP is we want to know what the association for the genetic component of that gene is. This is sort of like the missing predicted expression. So we can go to our reference data where we have all of the SNPs typed, including the missing SNP, and we can look at the association between those variants and the one that we want to, and the SNP that we want to impute. And so in this sort of idealized scenario, we see that the correlation between these SNPs roughly follows the same pattern. And in order to predict what we should have seen here based on our reference data, we basically just take a weighted combination of the GWAS statistics weighted by the correlation between these SNPs and the target SNP. And then the variance of that correlation is essentially very intuitively derived from the sort of variance of these weights, again, in the absence of LD. And so we have a final statistic, which is just the predicted effect size divided by the expected variance of that effect size under the null. Now, we don't actually live in a world without LD, but you can think of LD as just something that introduces additional variance. It produces more correlations between these SNPs. It produces some residual associations, and it also kind of shifts the observed effect sizes a little bit. But all we do to account for that is we need to model LD through this matrix of SNP-SNP relationships and basically just pull it out of the variance term. And so we adjust the variance, we account for the additional variance using the LD matrix here, and this is our final statistic. And so again, hopefully the intuition is helpful in that we're basically trying to infer an untyped variant, except that untyped variant is the expression of the gene that we're interested in. And so when we run this model, we can compare between what you would do if you just had the summary data, just the association statistics, and if you could actually do the individual level prediction, we see that the correspondence is almost identical. And it really should be identical if the LD is and the allele frequencies are perfectly matched between your two data types. So this is actually a nearly lossless analysis, and in fact, in some large studies like the UK Biobank, even when we do have the individual level data, we still do it this way because it's a lot easier. So we ran a couple of analyses to try to convince ourselves that this approach was actually honing in on real causal genes. So far we've just developed a method to connect variants to their target genes, but how do we actually show that they're doing something, that those genes are meaningful in some way? And one thing that we did, which I think is pretty clever, is we looked at a GWAS study of lipids that was done in 2010 and looked at about 100,000 samples. And we identified 25 TWAS genes that were significantly associated across the various lipid traits in these studies and didn't overlap genome-wide significant loci. So this is sort of novel loci that were identified just through the gene-based association. And then we looked at data from a later lipid GWAS study that was about twice as large. It was a superset of the same study, but it introduced another 90,000 individuals. And we see that for 19 of these 25 loci, they now become genome-wide significant. And this is highly significant, sort of no matter how you account for the distribution of these loci. So at the very least, I mean, this is not a direct demonstration that we're identifying causal genes, but it is a demonstration that the associations we see are significant, are predictive of what we will see as at the genome-wide significant level in future studies. And the gap between this 19 and 25, we actually, through simulations, think is consistent with just insufficient power in this study, and we presume that they will actually be observed at even larger sample sizes. So we've now taken this approach and moved beyond just analyzing gene expression traits, and we've thought about how to integrate other possible traits. And in particular, I'm talking about a paper that came out last year focusing on schizophrenia, and here extending the model to predicting not just gene expression, but also predicting alternative splicing from the Common Mind brain gene expression study, as well as incorporating chromatin activity that was measured by ChIP-seq in LCLs. And we're sort of essentially extending this model where before we just had the SNP and we were connecting it to the target gene that we believe is the susceptibility gene, but now we're also mapping the regulatory element that's associated with that gene through correlation to chromatin activity. And to give you an example of that, I'm just going to go back to the locus that I started the talk with, where we had a significant association and lots of genes to potentially target. And we performed a TWAS on this locus, and it implicated this MAPK3 gene as the most significantly associated. And we can actually do conditional analyses. We can ask, let's condition these SNPs on the predicted expression, on this sort of gene pseudo variant. And when we do that conditioning, all of the association effect goes away. And we can do this in the other molecular traits that we've identified. So when we look at the actual expression, the EQTLs for this gene, for MAPK3, and we condition on the predicted expression, we see again that all of the effect goes away. And because this is a specific locus where we also observed an association to H3K27 acetyl activity that was marking a regulatory element, again, we can look at the Chrome QTLs for that regulatory element, we can condition them on the predicted expression, and we see that the association goes away. And so we think that there's sort of more and more statistical evidence that at least the prediction of this gene is consistent with explaining all of the molecular genetic activity happening at this locus. And when we zoom out a little bit and look at how this breaks down in terms of all of the, at the time, 108 known schizophrenia loci from the PGC, we see that 26 out of the 108 have an association that was observed through alternative splicing, through a predictor for alternative splicing. 48 of those have an association through gene expression or splicing, and the majority of them are actually not the nearest gene. So, so sort of a simple heuristic would not have necessarily identified them for you. When we then introduce this layer of population chromatin activity, we see that for these 48 genes, a majority, 34, also are associated with nearby chromatin activity in independent data. And the majority of those are also not in the promoter. So again, the sort of simple heuristic of look at the nearest gene or look at the variant in the promoter of a gene would not have prioritized these potential targets for you. And the other thing that we see is that we're starting with a very, very large GWAS study, but then we're working down to smaller gene expression studies and even smaller chromatin studies, but we're still maintaining pretty good yield in terms of molecular mechanisms. And we think that that's because we're basically getting closer to the sort of the mediators of the this functional cascade. And so we need fewer samples to be able to see the chromatin association than the transcription association and so forth. So we've implemented this in a efficient and easy to use method, and we've sort of turned the crank on lots and lots of publicly available GWAS data, and we put this together in a web interface called TWAS Hub, where you can go and see the association statistics for hundreds of traits and a lot of different gene expression models, and you can kind of do these things on the website too and look at how the conditional effect operates. And it's one of the things that's sort of amazing with being able to operate on summary level data. So just in the last few minutes, I'm going to talk about next steps. How can we improve these methods, and what are some potential caveats to these methods? So the first critical question, and I think still an unresolved question, is all this time I've been talking about identifying the target tissue, but we also need to know I've been talking about identifying the target gene, but we also need to know the target tissue. We need to know the relevant tissue for that disease and the relevant tissue for that locus. And so in that schizophrenia TWAS study that I talked about, we actually looked at a number of different tissues, and these were all fairly large sample size studies. They all had at least 500 individuals with gene expression measured. This is from the common mind, and then also studies with adipose and blood expression analyses. And at the level of top hits, we actually did not see a whole lot of tissue specificity. So there were about as many loci that were resolved using expression predicted from brain as there were that were resolved from these other tissues, which we imagine are unrelated. When we looked at something like a polygenic risk score, where you don't restrict to genome-wide significant gene associations, but you sort of allow, you construct a predictor from every single association all the way down to nominally significant ones, and then we run those predictors out of sample into an independent schizophrenia cohort. We see that, well, first of all, as with disease architecture in general, it's very polygenic, so the more genes you introduce into your predictor, the better you do. But also we start to see this tissue specificity or tissue relevance emerging. So the expression models that were trained in brain were consistently more predictive out of sample than the expression models that were trained in these other tissues. And if you combine all of this together into a big sort of mega polygenic predictor, that predictor explains about a quarter of the heritability of schizophrenia, or the SNP heritability, sorry. And that predictor is largely explained by gene expression that was measured in the brain. So I think the story that's emerging is that the tissue where we expect things to be going on is where those mechanisms are happening, and we probably can fill this out, increase this proportion even higher by getting closer to the right cell type or to the right condition. But at the same time, you see a lot of residual effect in other tissues too, and these are probably just sort of echoing what's happening in the relevant one. And so if you don't have the right tissue, you can still get some traction, and it's not insignificant from other proxy tissues that are not necessarily related to the or not perfect. And again, it's probably the case that this brain expression measured in GTEx is also not really the perfect tissue, and maybe we need something measured during a specific stage of development or during a specific exposure. So we both want to be able to kind of leverage all the data we have and progress towards the more appropriate tissues. The other ways to kind of boost power for these analyses, besides just collecting more data, are to integrate the predictive models from multiple tissues. So again, GTEx is sort of a unique resource in that it's collected expression from many, many tissues, oftentimes in the same individuals. And there was a paper earlier this year, Hu et al. In Nature Genetics, that constructed TWAS predictive models that used all of that gene expression together and did something like a group lasso where they're penalizing these expression weights to be similar across the tissues. And they see, so on the right here is the percent gain in predictive accuracy by using all tissues together rather than just the focal one. And they see, I think, really substantial predictive gains. And we've played around a lot with using more complex models in a single tissue, and we really almost never do much better. And they're seeing really substantial improvements by integrating multiple tissues. So I think that there's a lot more that can be done to train better models by using external data the way that they're doing. The other approach is you can test multiple models at the same time. Instead of, up here, we're training with expression that was measured in 44 different tissues, but we could alternatively, and in some cases we're restricted to, only build a model in each one of these tissues and then combine the association of those models. And so there was some work that we proposed in the TWAS paper, and there was a paper from Barbieri et al. Earlier this year proposing a kind of multi-degree of freedom test where if you have a gene that's measured in multiple tissues and that's predictive, you can identify the kind of individual TWAS statistics for that gene. So here we've got one gene measured in 3 tissues. We've done the TWAS and identified the marginal associations, but then using the correlation between these predicted expressions, we can also perform a robust multiple degree of freedom test for whether there's an association in any tissue. And they've taken it a step further and performed some additional sort of decomposition and denoising on this data, so you're not doing, a 100 degree of freedom test here when you have hundreds of tissues, and that significantly improves power. And when they applied this approach to a large number of diseases, they see that if you just test with the relevant tissue that you expect, you get some results. When you test with all tissues, like marginally, if you just look at these individually, you get a lot more, and this is just because you're sort of incorporating EQTLs in other tissues that were not observed in this one. But then when you do this more robust combined test where you're jointly testing the association of multiple predictors, you increase the number of associated loci even further. So again, you can train better, but you can also test better by integrating different tissues and different models. Okay, and then the last thing that I want to talk about is also based on some work that came out literally in the past couple of weeks, but that's thinking about the limitations of this approach and potential ways to overcome those limitations. So there's a paper from Weinberg et al. A few weeks ago in Nature Genetics that sort of was a perspective on what TWAS is doing and what it is able to do and what it's not able to do. And I think the main conclusion from this paper is that there's several clear ways in which you can identify an association that's not the causal tissue. And this is to some extent expected because we're doing an association, we're going to be tagging lots of things, and causal inference is really hard. And so this paper does a lot to help understand how we can identify the non-causal gene. And so they break it down into 4 possible scenarios. So I think the simplest are that you're testing a gene which is the wrong one, but it's correlated or co-expressed with the right one. And so you'll see an association for the wrong gene just as you'll see an association for the right gene. So this is, I think, intuitive. And then obviously, because we're actually testing the genetic component or the cisgenetic component of that gene, you'll see the same thing, even if these genes are not very strongly co-expressed, but they have the same genetic driver, then this variant will go into the weight for the wrong gene, and it'll go into the weights for the right gene, and again, you'll see an association between these two. And so this is something to be aware of when you run a TWAS and you see 6 or 10 genes associated in a locus. It's possible that this kind of scenario is happening where you have an EQTL that's affecting lots of genes at the same time. And then there's somewhat more pernicious scenarios where you look at the wrong gene and it has actually a bunch of different EQTLs that drive it, but it sort of tags the GWAS association. And then the right gene is primarily driven by the GWAS association. And in this case, again, just the same way that SNPs tag other SNPs, you'll see the predictor for this gene can tag the association and appear to be significant. And likewise, you can have a false negative. So if there's an EQTL for the true causal gene, but you missed it because you are working in the wrong condition or in the wrong tissue, then you won't identify the true causal gene. And so again, I think this sort of breaks down to the fact that correlated genes will, as expected, come up as having correlated association statistics, and missing data or incomplete data will also result in missing or incomplete association statistics. And one approach that we've developed to try to mitigate this or to try to quantify it, and it kind of brings us all the way back to the first talk, is performing fine mapping, but at the level of genes instead of at the level of SNPs. And so around the same time, this paper of Mancuso et al. Came out presenting a method called FOCUS, which takes TWAS associations. It takes genes that you think are significantly associated, are genetically associated with the trait. And then in the same way that we would perform SNP fine mapping to identify the putative causal variant, you can look at the correlation matrix of the genes, you can look at the association statistics of the genes, and you can perform gene fine mapping and get a credible gene set that is prioritizing the genes that you think are the most likely, the strongest associations here. And we think that this moving more towards credible sets of genes and posterior probabilities of associations is the way to kind of quantify and try to get around the potential biases that come from just looking at one gene at a time. So in conclusion, I hope I've convinced you that in general, molecular phenotypes such as gene expression operate essentially like a complex trait. They are heritable, they have a very strong cis component of heritability, and they're predictable just the same way that complex traits are predictable. And what we've done is leverage that predictability in this TWAS method to identify associations to disease. We've also identified associations to chromatin activity that allow us to prioritize regulatory elements, although, as people know, the data from epigenetic activity is still substantially more limited. And then we're starting to see, as these methods are maturing, that we get a lot of associations in some loci. Not all of them are causal. Most likely, A very small number of them are causal, and we're developing methods to try to prioritize at the gene level as well. And all of this has been implemented in open source software that is easy to run, and it works with summary data that is in the same format as for LD score regression and some of the other tools that are used here at the Broad. And we've computed weights from a bunch of different expression studies, including the latest GTEx and TCGA, splicing variation, et cetera, and have a lot of scripts. So hopefully this is also something that you can use yourself. And with that, I'll take any questions. Thank you. Yeah.
Given what you said about the power of aggregating expression in multiple tissues, did you ever happen to ask whether just looking at brain itself, a very heterogeneous tissue, could be an okay predictor of non-CNS disease? Because you're essentially aggregating results of lots of different kinds of cells just in brain tissue.
Yeah. No, that's a great question. So let me think. So the analyses that we did were from the common mind, and that's all the prefrontal cortex. And part of the restriction here is that you need a few hundred individuals to be able to train the models. So I think that was what constrained us from looking at within brain subtypes. Yeah. Right. No. Yeah, I agree. So I guess I just don't have an empirical answer for how that actually works out. We haven't done it. We have these GTEx models, and in principle, like I could go and look, but I think we haven't seen clear evidence of combination, which specific tissues can be combined in a way that either improves power or decreases power. So far, I think everything that you've seen is really like this result, that if you just lump everything together, you do better. But there surely is a smarter way to lump things together.
Do we have any other questions?
Hi, I have a question about the interpretation of the results that you get from the TWAS. So you have a risk score for a gene for the transcription, and it is essentially a sum of weights that you then test for association with the disease. So if a SNP with a sufficiently non-zero weight is for whatever reason associated with the disease, you're going to get a significant result, even though that expression of that gene may not necessarily has to do anything with the disease. So how do you address that in your interpretation? Yeah,
that's a great point. So I think that's kind of touching on this scenario where this gene, this SNP isn't really contributing to this gene very much, but it is a GWAS hit. It has a really, really strong association.
But the GWAS hit could be for non-causal reason as well.
Yep.
So it's kind of double of a problem.
Yeah.
What you are showing is just part of it.
Right. So, the way that we try to do this statistically is through conditional analyses. So, like I think I showed in the previous plots, where we condition the locus on the predicted expression and see if any of the signal remains. So
and that would explain if that SNP contributes to the gene, right?
Yep.
But my question is mostly about if a SNP is associated with the disease for whatever reason, not necessarily a causal one. So it's not about the gene contributing to the, it's not about a SNP contributing to the gene, it's about a SNP contributing to the disease. So a SNP can be associated with the disease for whatever reason, many reasons, not necessarily causal. And if that SNP happens to have a large enough weight, your PRS will be significant.
Right. Yeah, that's true. Yeah, I don't think we have an answer for how to resolve it. I mean, do you think that the SNP is associated with the disease?
Yeah, so like when we have a locus, and that locus could be several genome-wide significant SNPs, doesn't mean that all of them are causal.
Right.
So essentially the PRS is kind of a test for association. It doesn't necessarily test the underlying hypothesis, even though it often gets interpreted this way. I was just wondering if you have an alternative way of dealing with that. Because you in a locus, you say you can have many when you genome significant hits, and
Yep.
none of them may be causal.
Yeah, no, I don't think that we can go much beyond just, there's a genetic association that is between this gene and the disease. It does not have to be a causal effect through this gene, but that something that is affecting this gene is also affecting the disease or associated with the disease. I think we need to do either like real experimental validation to distinguish that. We're trying to integrate other molecular features. So, if you see that there's also an effect on chromatin that gives you more confidence that this is a real cis-regulatory mechanism, you can look at other tissues, at other diseases and see if it's sort of concordant, and you can look at other populations. But this is all just ways of trying to get some other data to confirm the association. If you don't have that other data, I don't know statistically what you can do.
Have you ever tried to validate a TWAS result with the actual molecular data?
Yeah, so in this paper, in this paper, actually this locus, the MAPK3 association, we worked in collaboration with Nico Katsanis, and he showed some validation in zebrafish. So it's imperfect, but we took this SNP that was in a specific regulatory element and showed an effect on the gene that I think was consistent with the biology of this locus. We've had a recent study where we've looked at an effect that was only seen through splicing, actually, that was not seen through overall gene expression. Those are a little bit easier, like if they're actually in the splice region, it's a little bit easier to know what to do with them. It's not exactly non-coding anymore, and so we validated one gene through a splicing assay, and we've done recently in kidney cancer, we've done a CRISPR analysis that also showed an effect on a regulatory element and on a gene. And so I think actually the epigenetics is one thing that consistently has been very helpful to make us more confident about these loci. But that's, 5 years and 3 loci, and they're still all kind of vaguely validated. So that's the rate that things are happening.
Thank you.
Great. Thanks again to both of you. Let's
Thank
thank
you.
our speakers.